But what is this requirement for a DNA end? Remember that DNA is helical, so that it's not only going to have to make the proper base pairs, but it's also going to have to wind around that circular DNA once for every 10 or so base pairs that get made. And that's why you have to have a DNA end. If you don't have a DNA end, the reaction can go on, but you'll need an additional enzyme, an enzyme you've already learned about, and that's a DNA topoisomerase. And that will allow the strands to pass between each other and allow the helical form of DNA to occur. Now that you understand in schematic form the REC-A promoted strand exchange reaction, let's look at an in vitro reaction that occurs with molecules in an experiment in the lab. You have the single-stranded substrate, and you have the linear duplex, and then you add REC-A protein, and you'll get a molecule that looks like this. The circle is kind of rolling into the duplex, and you start making Watson-Crick base pairs here. And you also start twining around the DNA here to make the helical form. This is the strand that's getting extruded, and that will end up being the leftover single-stranded DNA. And when this circle has completely rolled into the linear DNA, you'll get a nicked circular product. So let's look at these products on a DNA gel. So at the beginning of the reaction, we have a linear duplex DNA molecule here on a native agarose gel. And the single-stranded DNA doesn't stain as well, but it migrates down here on the gel. So once the reaction started, you start seeing things happen right away. You start seeing DNA in this upper part of the gel, which represents all the different forms of these intermediates that are in different stages of the circle invading the linear DNA. But quite rapidly, after only 15 seconds, you see a large accumulation of the next circular product DNA, which is this DNA right here. And as you continue to incubate the reaction, you basically chase all the material into the strand exchange product. So if you think about this reaction for a minute, as you are continuing to incubate the reaction for a long time, you might get slightly puzzled, because the stability of these molecules and the stability of these molecules should be the same. So you might think that at the end of the reaction, you get a 50-50 mixture of these DNA products and these DNA products. But that's not what we see. We see that all the DNA is chased into the next circular DNA. That's because REC-A is catalyzing the strand exchange reaction. It is only taking the reaction forward. It does not take the reaction backward. And REC-A will stay bound to this product DNA for quite some time. And it's that stability of this end product with the REC-A bound to it that makes sure the reaction goes only one way. And so you get complete strand exchange rather than an equilibrium between the two types of products. Exchange is favorable and it's rapid. In vitro experiments, you can see extremely rapid strand exchange reactions where a REC-A-coated single-stranded DNA can find its complementary match in the DNA of a whole genome in just seconds. So now we want to go on and see how these powers of REC-A to be able to do strand exchange work in the double-strand break repair pathway.